thanks very much for organizing this whole event. This has been a great symposium so far. I'm very happy to be here to contribute. So yes, as Laura said, I'm going to tell you about human neuraminidases. So think of changing gears a little towards thinking about catabolism rather than anabolism, right? Not building things, but taking them apart in defined ways, right? So I'm going to tell you a story. I'm going to tell you a little bit of chemistry about how we built some tools for these enzymes, and then I'll talk a bit about applications that we think they're going to be very valuable for, and I'll show you some examples. And the great part about talking in a symposium like this is most of my intro has already been covered. So these enzymes, I think we all know, well, probably everyone knows the cartoons. Just to remind you, those cartoons have some chemical structure behind them. So the neuraminidases cleave neuraminic acid, which is this terminal residue that you see here. That can be found in 2,3 or 2,6 or 2,8 linkages. And humans have 4 different enzymes to do this one chemical transformation. And of course, the reason they probably have them is because there's many different substrates, right? I've simplified just one end of that substrate. I'm leaving out all the complex stuff like the protein it's probably attached to or the glycolipid it's attached to. But this is the transformation that they're all going to catalyze. Notice that they typically will liberate a galactose binding site, and I'll mention that again later. So humans have 4 of these neuraminidase enzymes. They're found in different subcellular compartments. They're found in different tissues. They have different expression levels in those different tissues. The ones that we know are at the plasma membrane are NEU1, NEU3, and NEU4. These are also found in the lysosome, because you can imagine these are shuttling back and forth. NEU2 is a cytoplasmic enzyme, and NEU3 has been reported to be at the nuclear membrane as well. Other general things we know about these enzymes, they prefer different substrates. So NEU1, 2, and 4 are known to generally, and I'll clarify that a little, modify glycoprotein substrates. They like N-links, unlike Warren, but and they also modify, the ones that modify glycolipids are NEU3 and NEU4. Now, NEU3 is the only one that, if you read the literature, the dogma is that NEU3 selectively targets glycolipids and doesn't modify glycoproteins. And I put a big star here because we already have seen many examples where that's not true. NEU3 does like glycoproteins in certain contexts. So these are just general preferences, but you can imagine that as enzymes that can modify these complex substrates, they can play roles in modifying what the makeup of those glycans are in a cell. So probably we typically think about the anabolic side of this, right? We're building glycans, we have to have these complex structures, and so sialyltransferases do this business. They start typically from a galactose residue that they're going to tack that sialic acid onto. But the enzymes here that I'm going to be telling you about today are really just going to be cleaving these down, right? And you could think of this as just a chop it up and throw it away kind of process, but of course that's not really what happens. When you take a sialoglycan and liberate that galactose residue, now what you've done is you've either removed a binding site for something like a Sialic, or you've generated a new binding site for something like a galectin, right? And so both of these enzymes are essentially modulating those 2 downstream processes that go off from here, right? And so if we don't understand both of them, right, we're going to miss a lot of activities. So my group is very interested in trying to understand the substrates for each of these enzymes. We've done chemical studies to try and understand them more clearly. I'm going to tell you mostly about the inhibitors that we've built for these. And the reason that we've built inhibitors is so that if we can block selectively each of these enzymes, we can start to understand their biological function. And before I leave this sort of opening idea, I want to remind everyone of this paper from back in the 80s from the late, great Werner Reuter that taught us that glycans are dynamic, right? So you have to remember that we don't just build these things and then they do their thing and then they go away. They're very dynamic structures. What you're looking at here, these are half-lives from radiolabeling of individual either monosaccharides or precursors that ended up as parts of the peptide. And if you look at the relative half-life of those species, right? So if you put the label in the protein, let's just normalize that to 1. If you put the label in the core mannose, it's about around half of the protein. And look at fucose and mannac, which eventually becomes sialic acid. These guys are really short half-lives when you compare them to the protein, right? And this is in a whole animal, right? So this is if you just feed those radiolabels to the animal and then look at the glycoproteins, this is what you see. So I think this is a really important clue that these things are coming on and off. They're changing, right? The protein core may last longer than most of the glycan. And of that glycan, some of these terminal residues don't last as long as the others, right? And so that probably has something to do with these catabolic processes. Okay, so let me tell you a little bit about the work we've been doing with these. So I'll give you one little piece of a substrate study that we did to try and understand these. We've done lots of other work in this area, but I think this is a more recent piece of data that we that we did looking at specific types of sialic acid. So this is meant to remind everyone that, we see the purple diamond all the time for sialic acid, but there's more than one sialic acid. There's sialic acids that have modifications in the 5 side chain, right, the glycolyl derivatives. There are sialic acids that have acetyl groups on them at different positions. The 9-O-acetyl is the one that's shown here. And one question you could ask yourself is, do the neuraminidase enzymes like all of these equally well? Do they process all of them because they're all there natively or not, right? Now, these are a lot less common than NEU5AC, the typical purple diamond. But we wanted to ask whether the different isoenzymes just have different preferences on these modified substrates. So we use HPLC for this. There's some data over here showing you individual peaks that we can assign to different forms of sialic acid. And what you're looking at over here are ratios for either the NEU5GC to AC, when we start with a complex protein that has both sugars in them. And then we just look at how rapidly they are cleaved from that protein. And what you can see here, we looked at a number of different neuraminases and we normalized to just an acid hydrolysis. But you can see that, for example, NEU3 doesn't really like that NEU5GC substrate at all. This bacterial enzyme also doesn't care for it. These others look slightly lower, but they're not statistically significant, so we can't say that they're really doing anything different. But at least some of these isoforms have a selectivity. If we look at the 9-O-acetyl, you can see here every one of the human enzymes doesn't like that 9-O-acetyl, to different degrees, of course. But this may actually offer a clue as to why you have those 9-O-acetyls, right? They might turn off the catabolic pathway here, or at least slow it down for certain substrates, right? And there's some important cancer biology behind or known about these 9-O-acetyl sugars, and so you can imagine that these could play a role in those diseases. Okay, so I'm going to switch gears a little and talk about tool development in this system. So we can make these enzymes, we can look at their activities. But one of the questions that we started with in this system was how can we turn them off, right? How can we control them and develop tools to target them? And when we started in this back around 2008, 2010, there weren't very many examples of inhibitors for these enzymes. So probably many of us know viral neuraminidase was already brought up. Probably many of us know Tamiflu over here, which is oseltamivir, or Relenza, which is zanamivir. So when flu season comes around, we hear about these to target the viral enzymes. And actually, when people look at this, one question you could ask is, these things, the viral enzyme catalyzes the same reaction as the human enzyme, so shouldn't these compounds also target that? Well, the answer is not really. Not very well, at least. So oseltamivir in particular, if you look at the human neuraminases, it's a terrible inhibitor. It's not very good at all. That's probably because it's lost all the hydrogen bond donors and acceptors that should be sitting out here in the glycerol sidechain. Zanamivir, though, keeps those, and it's still not all that great. It's okay. It's single-digit micromolar for NEU2 and NEU3. It's not very good for NEU1, and it's not that great for NEU4. So you will see people use these to try and target the human enzymes, but I want to remind you that this piece of data is out there. It's not just us. A couple of groups have pointed this out. And in fact, back in 2008, Kiso's group was the first to report a selective compound that could target just one of these enzymes. So they made a very small modification here. They put an amine at the 9 position, acylated that, and this compound, which they refer to as C9BA Dana, is selective for NEU1. I'm going to be referring to a lot of things that are based on this Dana core, which you can see that has elements in all of these compounds. This is really just sialic acid that's lost a couple of hydrogens here. And this mimics this sp2 hybridized carbon here. Sorry for any flashbacks to organic chemistry, but that sp2 hybridized carbon helps to mimic the transition state for that enzyme. Okay? So what we thought when we looked at the system was we need better tools. This is good. This is selective. But it's not very potent, right? And so we started a program a few years back now, basically starting from Dana, modifying it in selective ways to try and find new inhibitors. And so I'm not going to show you all of that, but I'll just give you a little flavor for how that was done. So what you're looking at here, this is a crystal structure. These are 2 structures overlaid. It turns out that there are 4 human enzymes, but only one of them has been crystallized, right? The cytoplasmic one, NEU2, is very easy to work with. It's very stable. You can crystallize it. You can get co-crystals. And so this blue mesh structure is that crystal structure that's been co-crystallized with Dana in the active site. So we thought when we started on this project that we would basically just take that structure, make a homology model to the other enzymes, and start thinking about how to target the active site. Right? This is pretty standard medicinal chemistry. So you can see here, if we overlay this gray structure as NEU3, a homology model, so this isn't real data. But you can see, for example, there's a glutamine residue sitting here in this blue mesh that's in the NEU2 structure that's missing in the NEU3. So you can imagine maybe you can put something big out here to target NEU3. And that was our very simplistic model of how we could attack this problem. We're not messing with anything out here. These standard interactions for most of the sialidases we're leaving alone. We're just going to start poking around on the outside of the molecule. All right, so that was back in 2008. We started doing that. And what you're looking at here, this is just a short update for me to point out that that model is still probably our best. These are overlays of AlphaFold. So when AlphaFold came out, I was all excited. I thought, well, maybe we can get some great structures. What you can see here, this is an overlay of our homology model that we made back in 2008 or so with the blue AlphaFold model. And the active site is right here in both of them, and they're pretty much dead on top of each other. So the stuff that's disorganized in the homology model is disorganized in AlphaFold. But I want to say AlphaFold doesn't solve other problems, but it doesn't solve our problem. So we still are essentially flying blind structurally, but that doesn't mean we can't find good inhibitors. Now, if we're going to screen inhibitors, we need assays. And this is actually one of the problems in the field. If you've got 4 different isoenzymes and you want to screen them, right, you have to be able to generate each of them and selectively measure IC50s or potency. Okay, so the way we do this, I'll just summarize it quickly. We produce NEU2, NEU3, and NEU4 recombinantly in E. coli. This is doable, but it has some problems. The proteins are not that stable. They don't stick around for that long, so you have to make them quickly and use them, and there's just some handling issues you have to deal with. NEU1, you can't do this at all. NEU1 has to be co-expressed with other proteins for it to be stable. And we've tried, it doesn't work. So if you produce NEU1 by itself, it doesn't work very well. You have to produce it in a mammalian cell that also co-expresses that other protein. Okay, so we express this in mammalian cells. It's not purified typically. These are purified and recombinant. And then to make sure that we're not misled by this NEU1 assay, because it can have other activities, we also go back and we confirm everything in homogenates of mouse tissue that we get from mutant animals. So we have a long-term collaboration with Alexei Pazhetsky's group at Montreal, and they have made these mutant animals that are NEU1, NEU3, or NEU4 knockouts, and also double knockouts. And we can do IC50 curves like what you see here, right? So in the wild type, these are just 2 compounds. One is selective for NEU3, one's selective for NEU4. Actually, this one targets both 3 and 4. So you can see in the wild type, they're both showing inhibition of some neuraminidase activity if we just measure general neuraminidase activity. But in the NEU3-4 double knockout, there's no inhibition. So that tells us it's at least selective for one of those 2. And you can see here with the NEU3 knockout that the red compound is targeting NEU3 better than the blue compound, and then here with the NEU1 knockout, where you've gotten rid of most of this excess neuraminidase activity, now you can inhibit almost all of it with the NEU3-4 selective compound, and the NEU3 selective compound isn't quite there. Okay? But these tools basically allow us to figure out what is the selectivity, what's the specificity, and without these, we would be lost, right? So we need these assays. I'm simplifying the years of work that went into getting them, but with those, what we did is basically make a lot of libraries, right? And so we have, at this point, we reported most of these in 2018. We now have a panel of inhibitors that allows us to target each of the enzymes, okay? And so these are all listed with their IC50s, and their selectivity is shown between one neuraminidase to another. So if it's very good at targeting NEU4, for example, its next best inhibition is 500-fold lower than that. Okay. So in this case, this is probably our best compound. It's 30 nanomolar active, 500-fold selective. Just one small group that we've attached to the 9 position here. The compound I just showed you that was 3,4 selective is a big greasy biphenyl hanging off of the 9 position here. And if we modify that compound with a guanidine, we can now selectively target NEU3 over NEU4. NEU2, we don't have great compounds. These are about micromolar, but they are selective. And we've got some good NEU1 inhibitors. So that original C9BA compound that I told you about, that's about 3 micromolar activity. We're now down to 50 nanomolar when we make 2 modifications of the sialic acid, or of the Dana scaffold. So we've got these tools, and now what we've really shifted into is a couple of things to try and improve these a bit, but also start to move them into asking some real biological questions. So we have our cartoonish models, so again, we have no structures, but the way we think about these systems is essentially based on the structure-activity relationships that we have. That we have a large hydrophobic pocket on some of these, and we're able to accommodate relatively long tails in NEU1, but that's not really true in these other enzymes. And so those differences in the pockets allow us to get selectivity. I always like to think of kinase inhibitors when I show this, right? It used to be, back when I was a grad student, right, finding a selective kinase inhibitor was a big deal, and people thought it was going to be really challenging. The same can be true for glycan isoforms, right? We have to start thinking about ways to target these selective isoforms the same way that the kinase community has done that. What we're currently doing in the inhibitor space, I can't tell you about a lot of it because some of it's sort of protected by IP or is in the process of being protected. But we're essentially working to do, I would say, two major focus right now. So one is we're building better models. So we now have a data set of many inhibitors that target each of these isoenzymes. And so we're trying to use that data to build more quantitative models. So we built Pharmacophore models for each of the enzymes. We've built QSAR models for these. These QSAR models are essentially allow you to predict activity based on structural factors. And we've essentially built these models. Now we're in the process of testing them and validating them as a way to make better inhibitors and to do it with a little bit of a computational flavor to it. We're also working to improve the bioavailability of these compounds. These are all carbohydrates. They're all polar. They're not great drugs, right, as they start. And so we need to make them better. And so we have a few strategies that are in the works. I can't tell you about them yet, but we're moving towards making these much more useful as therapeutics. Which is not to say we can't use them as therapeutics now, but mostly in the lab. So let me move. I'm going to change gears from chemistry and talk about biology for the rest of the talk. And what I want to tell you about is a couple of vignettes about ways we've used these inhibitors to learn something about neuraminidase biology. And actually, this touches on something Warren already brought up. So let me just orient you first on atherosclerosis. So we probably all know a bit about atherosclerosis. What you know in the popular press, right, is that there's good cholesterol and bad cholesterol. If you remember, bad cholesterol is the LDL, or the low-density lipoparticles. And I want to talk a little bit about the structure of those LDL particles, because I knew nothing about these at the start of the project, but they're really pretty fascinating. So one of the things about these particles is that LDL levels, of course we know, are elevated in atherosclerosis. People develop atherosclerotic plaques. That's usually thought of as going through the LDL receptor, which pulls that LDL particle and all of its associated lipids into a macrophage, and that forms foam cells. Those form the lesions. Okay, so fats essentially from LDL get deposited into the plaques through the macrophage. But it's been known for a long time that a couple of important glyco facts here is that the LDL particle itself is heavily glycosylated. It's got tons of sialic acid on it. I'll show you a bit of the structure in a second. So first of all, it's got glycans on it. But the other thing about this is that it's been known that atherosclerotic patients with atherosclerosis have elevated sialic acid levels. They have different levels of sialic acid in the atherosclerotic lesions themselves. So there's clearly some element of glycobiology here, but what's going on? So one hypothesis is that, and the hypothesis that my collaborator Alexei came to me with, is that what if these particles that start off silated become desilated, right? They now have galactose residues expressed on them. Now they're substrates for the asialoglycoprotein receptor. And as Warren mentioned, this is typically thought of as being on hepatocytes. It goes through the liver. But that's not strictly the case. So Alexei's group has actually shown that receptor is also on macrophages, right? They've got tons of asialoglycoprotein receptor on here. So this is one more way for these to get in. And so you could imagine then, if neuraminidases are somehow regulating the silation of LDL particles, they could be regulating this uptake process. Okay. So we decided to start looking at this in vivo. And what you're looking at over here, these are sections of mouse aorta. This is from an ApoE knockout animal that develops atherosclerotic plaques regardless of what you feed it. And the plaques are stained with this red dye here. Okay, so the ApoE mouse looks like this, and this is just individual sections, but there's quantitation for multiple down here. If you look at a NEU1 knockout, or a NEU3 knockout, or a NEU4 knockout, the question that we had was, does this enzyme matter to this process? Do you have a change in atherosclerotic plaque formation if you get rid of the neuraminidases? Because that would imply that maybe this LDL silation matters. And there's other mechanistic work to back this up as well, but that's exactly what we observed. So for NEU1 and NEU3, we see a decrease in lesion size. These are all double-blinded, so we trust these results. This NEU4 result is very spread out, so we're not sure if NEU4 is really involved yet. We can't say from this data. But this did give us the indication that the neuraminidases could be involved in this process. So of course, the obvious question that we would want to ask is, if the enzymes are involved in this process, could you block it? Can you block it pharmacologically? Because that would then also build the case that this is a relevant step in the pathway. So let me just tell you a little bit about ApoB, just for two reasons. One is just that it's a cool protein. It's huge. It's about 4,000 residues. What you're looking at here is just the primary sequence mapped out numerically with different domains color-coded. Each of these arrows are predicted and link sequons. And what you see here is the data from a 2005 paper that looked at where are the N-link glycans in this protein. And so they identified, I just put purple diamonds at positions where there were glycans that contain sialic acid. You see many of these are populated in the human ApoB. When we did this experiment, so we basically took human, isolated LDL from human blood samples, and then we did the same experiment using proteomics, and we also observed many of these sites, not exactly the same, but many of these sites sialated. And then if we treat that protein with NEU3, in vitro, we lose most of those glycans. Okay, we lose the sialic acid part. So that told us that NEU3 could modify these. This is just some of the data to show you that. So you can see like these heavily sialated peaks, like 11 here goes down, 7, 4 comes up. So these asialo and these lower sialated peaks pop up, right? And so we're able to show that this can happen at least in vitro. There's lots of other in vivo validation of this, but I'm going to move on to the pharmacology. So you're looking at here these micrographs again. So we're looking at plaque sizes, again, double-blinded. We take those mice and we treat them with our compounds. We treat them anywhere from 1 to 10 mg per kg. They're getting an injection of this once a day, I think every other day for a couple of weeks, leading up to when we sacrifice them to analyze this. And you can see that NEU1 inhibitors like C9BA, the one that was reported before us, and our compounds here all block atherosclerotic plaque formation. Same for this compound that targets both NEU3 and NEU4 together. This one just has a different control, so we have to do it in a separate run. So we're really excited about this for a couple of reasons. It shows you that the glycosylation is important in this pathway, but also that we could interfere with this in a completely new mechanism of action. So as I said, this is going to be a couple of vignettes, and I'm going to switch gears to more of an inflammatory or immunology story. And one of the questions that our lab has had for a long time is that we know, so Hans gave a great introduction this morning to glycans in some immunological systems. We know that glycans like Sial-Lewis X are important for inflammatory response. So you can imagine then that anything that cleaves sialated ligands could also modulate those systems, right? So one of the questions we've had for a long time is, do these enzymes participate in inflammation and recruitment of leukocytes in any way? And really, there hadn't been any systematic studies of this. So, so a PhD student in my lab, Amran, went to Alexei's group and actually worked with them and their knockout animals. And we just asked a very simple question. If you take a model of inflammation, right? So in this case, what we did is we take the mouse, we inject a sterile pocket of air under the skin, and then we can inject something like LPS or just saline into that. And then we wash that out after some period of time and ask how many leukocytes went to that site. Right? So this is just a model for a sterile mimic of a site of infection. And the question is, do you get a different number of leukocytes depending on which neuraminidase you lose? Okay? So what you're looking at here, these are numbers. The gray dots are when they've been LPS treated, and the white are saline. Here's a normalized LPS data over here. And these are just cell counts from those pouches. When you see as wild type, of course, you see an increase in leukocytes, because that's what should happen if it's a healthy mouse. But as soon as you go to NEU1, and it's probably clearer to see here, you lose that response. The NEU3 knockout, it's a little noisier. It's probably not really that different, but we've lost statistical significance here between these two. But in the case of NEU4, we see a huge increase. We see a huge amount of leukocytes go into these pouches due to LPS stimulation. The 3-4 double knockout shows the same. So this illustrates another theme I want to convey, is that these two isoenzymes that do the same chemical transformation have the opposite biological effect, right? And if you just did, let's say you had an inhibitor for all 4 of these enzymes, you wouldn't be able to find that, right? You'd be lost in the weeds. So we're pretty excited about this. We've got some more data on this, but I'll just touch on a little of it. You want to know what the mechanism is, right? So why would NEU1 do one thing and NEU4 do the other? Now, there's probably years of biology to do in this, but here's just one experiment that I think speaks to what some of the mechanisms probably are. So what you're looking at here, this is a transmigration experiment where we take bone marrow-derived macrophages from the mouse, from the knockout animals, and then we ask, how well do these macrophages transmigrate towards a chemoattractant? And we do this on a beta-1 integrin substrate. So it's a membrane that's been permeable or treated with a beta-1 integrin substrate. And then we just ask how many cells get to the other side, right? So this is meant to mimic the transmigration process of a leukocyte that's captured and is trying to get to a site of inflammation. So remember that the inflammatory cascade, this is just a cartoon from this 2007 review, involves a lot of different adhesion molecules and a lot of different steps, right? So probably many of us know the selectins and the rolling step, right? So the selectin and PSGL1 type interaction, this depends on a sialic acid residue being in the right place, right? The cells then move on to undergo slow rolling, firm adhesion, and then transmigration to get to the site. There's a lot of different adhesion molecules that are involved here, right? So there's LFA1, which is a beta-2 integrin, which I hope to tell you a little bit about. There's also a beta-1 integrin, the VLA4, which interacts with VCAM1. So there's a lot of adhesion steps that could be involved here. So that's clearly a question that we would want to ask is the adhesion of any of these cells modulated by these enzymes? Okay. So in a transmigration experiment, what we're asking is, do we disrupt that particular adhesion interaction in any of these mutants? Okay, and the answer is in NEU1 at least. The beta-1 integrin adhesion process has somehow been disrupted. Now maybe the beta-1 integrin isn't getting where it needs to go, maybe it's misfolded, or maybe it's got a different conformation because the glycosylation on it has changed, right? So these are heavily glycosylated, all of these adhesion molecules are glycosylated. And so we don't know what exactly is happening, but we at least have a lead here for NEU1. NEU3, on the other hand, goes in the opposite direction. So again, telling you that these have different functions. And NEU4, while it had a completely opposite effect on leukocyte recruitment, doesn't do anything to this particular step. Okay. So there's a lot of complexity here. There's a lot of context-dependent effects. And we really have to dig into each of these to probably understand them well. But this is a great starting point for starting to figure out exactly what's going on. Now, so I want to continue to talk about integrins, but I want to tell you a little bit about, I want to give you maybe a simpler experiment to try and illustrate this point about selectivity. So for a long time, people have thought about adhesion molecules like the integrins, which this is just a cartoon here of a beta-1 integrin binding to fibronectin as its ligand. The glycolipids that are present in the membrane can modulate their activity. So for example, lactosylceramide is well known to change the amount of integrins at the surface, change their shuttling to the surface, and you can imagine those. The idea here is that the glycolipids can regulate the integrins. And so if you can watch cell migration, this is just a two-dimensional cultured cell assay. If you look at the migration, you should be able to see if there's a change here in the rate of migration or how much of those cells get into that space. So this is often called a wound healing assay. It tells you a bit about how those cells are going to stitch themselves back together using these adhesion molecules. And so the question we started with was a very simple one. If we have inhibitors that can block individual isoenzymes, can we understand which of these are involved in integrin migration? We've done this a couple of different ways, and so Amran, again, did one version of this, which is more of a plate-based assay, but it's the same type of question. What you're looking at here is just the rate of migration normalized to a control. This is just a positive inhibition control. And then we just treat with an inhibitor of one of our enzymes. Okay, so this is a NEU4 selective compound here, and you can see that this has a dose-dependent inhibition of cell migration for that beta-1 integrin. All right, great. So now you would think other inhibitors should do the same. Turns out if you take the exact same cell type, exact same assay, but now you inhibit a different enzyme, NEU1 in this case, now you get the opposite result. So now you have a pro-adhesive molecule. You're driving these cells to migrate faster by targeting a different enzyme. Okay? And again, if you had a compound that was not selective, you'd probably have missed this, right? So this is just here to illustrate that selectivity can be very important for pulling this biology apart because you've got a lot of overlapping activities. Okay, and just to drive this home, I won't go through all this data, but this is the same experiment just shown with a couple of different conditions. And I'll just call your attention to this experiment here. So this is Dana. So Dana is just our generic, our pan-selective inhibitor, right? And this compound it's reasonably active, but you can see here's the dose dependence going up to 1.5 micromolar of this compound. So we see a little bit of an effect. It's a bit of an inhibitor, but not very much, right? Whereas our NEU4 compounds, these are much better. Okay, but one question you should ask is, all right, so if they do different things, do they compete with each other, right? And the answer is yes. So here what you're looking at, this is compound 2, which is that C9BA compound I mentioned. It's a NEU1 inhibitor. That's pro-migratory, so these cells are moving faster. So what happens if you take that NEU1 compound and you mix it with this guy over here, compound 5, which is a NEU4 inhibitor, right? So that one was an inhibitor by itself, which you can see over here. But now when I mix them together, I get no effect at all. Okay, so again, if you don't have selective strategies here, you could completely miss this. If you tried to study this with Dana, you probably wouldn't be able to figure out what's happening because you've got multiple overlapping activities. So I'm going to stay on the transmigration theme here, and I'm going to talk a little bit about, this is still a beta-1 substrate, but again, another context-dependent effect. It's a theme. So a similar experiment to what I showed you for the bone marrow-derived macrophages. So we seed cells into a top chamber, we have a chemoattractant in the bottom, and we put some kind of substrate in the membrane. And so in this case, it should be a beta-1 integrin-mediated process. And we're using T cells from humans here. These are cultured T cells. And we can talk about better models for this too. But if we just do siRNA knockdown of these for NEU1 and NEU3, we see that it's a NEU1 and NEU3 modulate the process. So they would act, if you block NEU1 and NEU3, you should get inhibitors here. So what if we do this pharmacologically? Okay, so again, if we come in with our NEU1 inhibitors, NEU4 inhibitors, or NEU3 inhibitors, what we see is essentially in all cases for the beta-1 integrin in a T cell, so the cells I showed you before, those are epithelial cells, right? Now we're looking at a T cell. Each one of these has dose-dependent inhibition. Okay, so still with beta-1 integrin, but in a different cell type. Okay, so now we're not seeing opposite effects between NEU4 and NEU1. We're seeing the same effect. Okay? So probably because the substrate's different, right? Or the amount of expression is different, or something is different here, but we don't know what necessarily. But both enzymes here have the same effect. And now, so we're staying in the T cell theme. We're currently working on, so Amran's focusing on this problem as well. We are trying to understand beta-2 integrins next. So beta-2 integrins are selectively expressed in leukocytes and white blood cells. So these should be much more important for inflammation, or they should be exclusively important for inflammation, whereas the beta-1 integrin could have lots of other biological effects. And so one of the experiments that we're doing here is to just take this is a FACS assay. We take a bead, we put the ligand ICAM1 on it, and then we just measure binding. Okay, and so what you can see here in this FACS assay, we see binding on this axis. So when we see fluorescent beads stuck to the cell, we know that they're active to bind ICAM1. But we've mixed this actually with one other component here. This is a viability dye. So these cells are also stained with a viability dye. So if you see them in either of these two quadrants, that means that those cells are also dead, or at least have permeable membranes. And one of the really interesting things that we've come across here is that it looks like we can see changes in the viability for these cells. So let me emphasize, the compounds themselves are not toxic. Okay, but if we look at proliferation out a couple of days, right? So we have to culture them for 2 or 3 days before we see this. The amount of proliferation changes dramatically between the control and the inhibitor treated. So we think this is actually blocking proliferation in these cells, and this is probably because these are, this particular T cell model. This is a cancerous cell line, right? This is an immortalized transformed cell line, and so we're probably disrupting what we think is likely to be the EGFR pathway that keeps those cells alive. Right? And so not only do these cells have effects on adhesion, but they also probably have effects on proliferation. Okay, so we're starting to pull this story apart. And so all I'll mention to you is that Dana here actually increases binding to LFA1, so the beta-2 integrin, whereas compounds that target NEU1 seem to block it. And NEU4 also seems to enhance it. So this looks like another situation where we have opposite effects on one particular integrin in this cell. By the way, we've done beta-1 integrin in the same cell, and it's a different effect entirely, right? So it's very context dependent depending on which cell you look at, which interaction you look at, and likely which tissue or organism you look at. So I'm going to wrap up there, and so I'll just my main conclusion here is that neuraminidase effects are absolutely context specific, like much of glycobiology. And just some of the examples I showed you today, right? So in inflammation, we see NEU1 and NEU4 are opposite. In 2D cell migration, we see that NEU1 and NEU4 are also opposite the other way. In transmigration with a beta-1 integrin, at least in some cell types, this seems to all be the same type of effect. And in atherosclerosis, we've found so far that NEU1 and NEU3 all are positive regulators in that system. Okay, so I think this is mostly just a pitch that we should be digging for more of these examples in different systems. We're starting to look in pulmonary fibrosis to look at the role of NEU1. We've done some collaborative work on that. We've done a little bit of work in cancer on NEU4 in this, and we're currently working on platelet clearance because we think this is also an important component of platelet clearance because silation is important in those. Okay, so I'll just wrap up there, and I'll thank all the people that did the work. I should acknowledge that I'm on some patents for these compounds. I showed you a lot of data from Amran, but people that made compounds along the way, so Tianlin and Nahoko made a lot of the compounds that we used to determine these. Earlier work from graduate students in the group was critical for that. This work has been largely supported by GlycoNet. We're now supported by IRCOR and some NSERC funding as well. So I'll stop there, and thank you very much for your attention.